Good morning. We are here at the Collins Institute, and thanks to No Foundation for organizing this event to make some dissemination about the Meniere disease research program that we are conducting now at the University of Sydney. My name is Antonio Lopez-Escamez. I'm professor of Meniere disease and neuroscience at the university, and it's my pleasure to share you with all of you our work. But first, I'd like to recognize and pay my respect to the elders, past, present, and emerging, of the land of the Universities of Sydney campuses stand for. For thousands of years, they have shared and exchanged knowledges across innumerable generations for the benefit of all, and we are all very happy to work here in this land. And the first thing I'm going to do now is to present my team, because this is really the most important thing when you are trying to set up a research program, to have the right people and to have all the resources that the University of Sydney is sharing with us. So we have now 3 postdoctoral researchers, Viva Patil, that will speak later about epigenetics of Meniere disease, the way that we are doing, which is a new approach. Also, Christina Markova, which is speaking, she's working about tinnitus research, which is other of the areas that we have. And Prathamishna Daponia, we will be talking about the mouse model that we are trying to develop for Meniere disease. But we have also a research assistant and honor students and a set of clinical collaborators. That this is just a very short list of people working in Sydney, a different hospital. But we are growing, and we are having new people working with us in different states in Australia. So just to start, I would like to make like a very short overview of what is Meniere disease and where is the location of this condition. So the inner ear is located in the temporal bone, in the human temporal bone. And what happens is that it's a condition that involves the anterior and the posterior part of the temporal bone. So in the anterior part of the temporal bone, we have the hearing organ. In the posterior part, we have the vestibular system, which is an organ that receives the information of the position of the head, the position of the body. And what happens in patients with Meniere disease is that they have loss of hearing, and they have also vestibular problems, and they experience dizziness, and they experience vertigo attacks. So to understand what happens in this condition, we have to know that there is a bony labyrinth, and inside this bony labyrinth, we have a membranous labyrinth. And this membranous labyrinth separates these 2 parts of the inner ear, the anterior part and the posterior part. And it's a very complex structure that includes the hearing organ, as I mentioned, and 5 vestibular organs, the utricle, the saccule, and 3 semicircular canals. And these are in charge of collecting the sensory information, the information about the movement of the head, the linear and the angular acceleration of the head, and coordinate this with the position of our eyes. And this is how we can have a reference of the position of the head, the position of our body. What happens is that in Meniere disease, we have an accumulation of this fluid inside the red part of this membranous labyrinth, and this accumulation leads to a progressive disorder. So what is Meniere disease, in fact? So Meniere disease, first, is not a single disease. This is very important to have in mind. It's a syndrome. So that means that we have several symptoms that includes vertigo attacks, progressive hearing loss that usually involves the low and the mid frequencies, as you can see in the audiogram in the right part of the slide, but also patients experience tinnitus, so noise in the ear. These episodes of vertigo are usually associated with tinnitus. This is a very clear symptom. It's not that the patient experience the symptoms always in the same way. So some patients may start with only with tinnitus, some patients may start with the vertigo attacks, but at the end, over time, all the symptoms tend to overlap in the same episode. And this is how we establish the diagnostic criteria. Meniere disease is not really a common condition because it's observed usually in less than 1 in 1,000 in the population. The onset occurs in the 30s, in the 50s, among that. But the issue with the condition is that it may become bilateral in between 25 to 45% of cases. Usually, the condition starts in 1 year and in about 7 to 10 years in both the second year. But as I have explained, this is not a very homogeneous condition. We have some patients that also have migraine, some patients that have another autoimmune disorder, some patients that also show an allergic profile, so they have sensitivity. But this is something that is not observing all the patients. And we have a set of patients with different set of symptoms, and this is why we need very clear and detailed clinical information for that. And for these reasons, to conduct genetic and clinical research, we need to use the same language. And this is very important because in the last century, different definitions across the world were established for Meniere disease. So what a Japanese doctor understood as a Meniere disease was not the same that can be understood for a European doctor or even an American doctor. The Varani Society, which is an international society of neurologists and otologists and also neuroscience, scientific and doctors, was able to establish diagnostic criteria for Meniere disease according to clinical symptoms. Definite Meniere disease is defined when the patient experiences episodes of vertigo with a specific duration. And these episodes of vertigo are usually associated with low to medium frequency sensory nerve hearing loss. So not any type of hearing loss, not any type of deafness is really associated with Meniere disease. And the relevant thing is that we have to document the hearing loss in close association with the episode of vertigo. That means before, during, or after the episode of vertigo. Another important feature in the definition is the patient has to experience tinnitus, hearing, or aural fullness. So fluctuation in the oral symptoms are important in the same year that the hearing loss is demonstrated. And of course, other more common causes of episodic vertigo has to be excluded. And I'm thinking about benign paroxysmal positional vertigo, or even some vascular risk factor that may be important for because they're very common, particularly over 50 years old. So the thing is that we have a set of patients with different symptoms. Not all the patients has exactly the same symptom, and we perform clinical studies in large cohort of individuals with unilateral and bilateral Meniere disease, and we were able to identify 5 clinical variants. So the most common group is the patients that only have symptoms in the auditory and in the vestibular system. So patients that only have hearing loss and patients that only have episodic vertigo, this is the most common situation. However, there's also a group of patients that also have another autoimmune disorder, being the most common, a thyroid problem. So autoimmune thyroiditis is very common in Meniere disease, but other conditions could be that rheumatoid arthritis, psoriasis, and other disease in this spectrum. And this is probably a different subgroup with a different etiology of patients. But also, we were able to identify a cluster of patients that, in addition to Meniere disease, they report to have migraine. And this is also relevant because we know that there is a close association between migraine and Meniere disease from epidemiological studies. And currently, we think that migraine could be a trigger of the Meniere disease episodes of vertigo. A much more relevant group is familial Meniere disease. It has been known for a long time that families may have a relevant role in the etiology of the condition. And also, some other patients first develop the disease in terms of hearing loss, and over time they develop the episodes of vertigo. So as you can see from these two studies, Meniere's disease is not a single condition. What they have Meniere's disease in common is that at the end, all patients have an accumulation of endolymph within the cochlear duct, within this membranous labyrinth that I explained before, that involves the anterior and the posterior part of the temporal bone. And this accumulation may explain the hearing loss progression. So that's what happens. That's the reason that once you have an early damage in the ear, the accumulation on endolymph makes that patient start to lose their hearing, and that's why they cannot recover and they don't have any response with treatment regarding to the hearing. So keeping that in mind, we have been able to demonstrate some of the molecular mechanisms that could be involved in Meniere disease. And that's what I would like to remark again, that Meniere disease is not a single condition. We have a clinical spectrum, we have a clinical syndrome, and we already know that probably there are different mechanisms leading to the condition. So what we see in the clinics is probably the result of different responses that we have in terms of the immune system. So the immune system is probably very relevant in the maintenance and in the progression of the disease. And we have been shown that the immune system is also different in different subgroup of patients. So according to that, we have a group of patients that have an autoimmune disorder, that this is very easy to identify because they have been already diagnosed, as I mentioned, with rheumatoid arthritis or maybe with a thyroid disease. And this could be related, and maybe the clinical manifestation that the patient shows at the inner ear is only the result of this systemic process. Another type of immune disorder that we have found is autoinflammation. What means autoinflammation is an increase of certain level of protein called cytokines, particularly cytokine called interleukin-1 beta is responsible of autoinflammation, and this is like a very systemic and persistent inflammatory status that makes that the patients have a chronic condition. However, we have also been able to identify a third subgroup of abnormal immune response that is called type 2 inflammation, and this is associated with a different set of cytokines that maybe has a close relationship with allergic response. And these patients may benefit from some specific therapy against allergy, and these patients can be also identified for a persistent elevation of an immunoglobulin E. So this is something that can be done in the clinic, and unfortunately is not a routine examination in all patients with Meniere disease. And the last but not least, and this is a great part of our research program, is the familial Meniere disease. We have been able to identify, as I mentioned, that autoinflammatory and autoimmune disease are different mechanisms, and both of them can be treated with steroid, but they involve different type of cytokines. But familial Meniere disease is a different entity, it's a different mechanism. We know from epidemiological study that Meniere disease shows a familial aggregation. In European descendant population, around 8% of the patients have another relative in first degree. So they have a sibling, they have also a parent with the same condition. And we have used this strategy to study families using exome sequencing, which is a technique that is able to read the coding region of all the genes in the entire genome. And the first gene that we reported was published in 2015, and we identify a family with 3 women in consecutive generation with Meniere disease, with the full spectrum. And 2 genes were associated with the family, and that were the genes DTNA and FAN133A. And so far, we have been able to demonstrate that there's a number of genes, and now around 15 genes has been already reported, being the most common of them, OTOG, that encodes for the protein autogelin, but also myo7A and TECTA are genes that has been described in multiple families. So we have now several genes that have been reported in several families, which is a very strong argument for a genetic condition. We have also several genes that have been reported only in one family, but most of the families remain still undiagnosed because there could be genes that are already known now because sometimes the families are small. And we are working on that, and this is part of the research project that we want to conduct here in Australia, because we have an idea that all these gene encodes for proteins that are located exactly in the interaction between the hair cell stereocilia and overlying structure, which is called the tectorial membrane. As you can see here in the organ of Cori, we have these hair cells, these 3 rows of green outer hair cell, and we have this blue inner hair cell. So these hair are responsible to make the generation of the nerve impulse when the stereocilia are tilted regarding the tectorial membrane. And within that protein that links the stereocilia to the tectorial membrane are essential proteins in Meniere's disease, and this is what we have found. And I'm showing this slide to give you an idea of the complexity of this structure. There are already more than 1,000 proteins in the stereocilia. And the initial discovery, and this is what's the most surprising thing, is that we have found that most of the familial Meniere disease has a recessive inheritance. And the main gene, as I have explained, is auto, that encodes for a protein called autogelin. And this is a structural protein in the tectorial membrane that links to the stereocilia. Our study was able to demonstrate potential mutations in the auto gene in 14 families. However, we did not find that the rest of a sporadic patient has the same type of mutations. So what we have found is that auto is a recessive condition. So auto gene, the parents are carrier of the mutated allele, and the program, the patient has initially an sporadic Meniere disease. So because the patient is not aware that their parents are carrier. And this model of recessive inheritance has been demonstrated in 4 unrelated families with Meniere disease. And we have found that they share this couple of variants. So this model is called compound recessive inheritance, but the parents has no symptom, and the patient develops the condition. So these are the 4 families that we have already found. And as you can see, not all the family members develop the disease. And this is something that, for this reason, we think that we have to start to do genetic testing in patients with Meniere disease, even if they don't have any relative with the condition, and particularly if they develop the disease when they are really young. So patients that start with Meniere disease symptoms below 35 are probably the best candidates for genetic study. So just to give you a summary idea of what we think it could be the molecular mechanism and the cellular structure involved in the pathogenesis of familial Meniere disease, we think that this mutation that we have found in auto-TECTA and MYO7A result in a very fragile inner ear. So these structures are probably very sensitive to sound. And increasing acoustic pressure or another environmental factor could be the trigger for the Meniere disease phenotype. So that means that we have an opportunity to make the genetic diagnosis in a very early stage of the condition, and even we can anticipate which are the carriers of the mutation that can have a high risk to develop the disease according to this potential trigger. So just to summarize, what is the outcome of our Meniere disease research program here at the Collins Institute at University of Sydney? We are planning to conduct a large genomic study in Australia in patients with Meniere disease to investigate the inheritance. And why Australia? So we have here a cultural and very diverse population that include individuals with East Asian and South Asian origin that have never been investigated in Meniere disease. And we have the hypothesis that this particular individual may have a different genetic structure and maybe there are new genes to be discovered. Our program also includes the study of the genetic structure of severe tinnitus. We have conducted several multi-center studies in Europe, and we have found that there are some mutations, some rare mutations, again, is some just associated with severe tinnitus, and we plan to continue this study here. But the novel approach that Dr. Viva Patil and Dr. Pratamish Nadapunia will try to explain are the epigenetic factors that have never been investigated in details in Meniere disease. And we are going to design and try to test a real mouse model of Meniere disease according to the mutation that we have found in the families in Spain. And this mouse model will be the first humanized mouse model with the same sequence that we have found in the human studies to demonstrate that noise could be relevant and to demonstrate that this is a fragile strata that can explain the phenotype. And finally, and not the least, we will continue investigating the immunophenotype of Meniere disease. We have described it, as you know, several subgroup of individuals, and these patients are particularly, this approach is very promising because we can probably target some biological drugs that can modify this immune response that I've been trying to summarize. So now we are recruiting, and we are recruiting patients at the Collins Institute. We are recruiting patients in Sydney, but this is just the beginning. We would like to remark that we are looking for patients with a confirmed diagnosis of Meniere disease, initially with 2 years of follow-up, because sometimes the symptoms are not very clear. We are looking for the genetic study for patients that start with the condition before 35 years old, but we are also open to other patients for immunological study without this restriction. So we can also collect older patients. And we are interested in family, so patients with a familial history of Meniere disease, that means at least 2 cases in the same family, or one relative with auditory symptom, because one thing that we have already observed is that some of the families have relatives with only hearing loss, only episodic vertigo, so some incomplete phenotype, but at least one of the individuals has to confirm the diagnosis of Meniere disease according to the clinical criteria established by the Barani Society. So the goals of this research program, as I mentioned, is to identify new genes in the Australian Asian participant. And we are also planning to to generate a global research reference database, which is going to be very important because this will be an open resource at the University of Sydney that all researchers and all the doctors in the world have free access to that, and they will be able to use the genetic information from any patients around the world and compare the data from their respective patients with the reference data that we are planning to generate for many diseases. And to demonstrate that our approach is something that we can implement in the health system, we are going to start a randomized clinical trial here in Sydney, in the hospital, and this is one of the goals also of this Meniere disease research program, to change from the clinical point and to change the diagnosis to a more precise genetic-based diagnosis. And finally, just to mention that we are also under the umbrella of the Knopf Foundation. We have a website, and we really appreciate if you want to have more information about our research program, and we are also open to donation to our work, because this is really a long-term project. We're trying to set up the reference lab in Australia for many years. Thank you so much for your attention, and I will now give the opportunity to talk so you can listen to Dr. Viva Patil. She will be talking about epigenetic.
So good morning, everyone. I'm Dr. Viva Patil. I'm an epigeneticist working as a postdoctoral researcher in the Meniere's disease lab. So we've just had a really nice introduction about Meniere's and how it occurs. So today I was going to give you a brief introduction to my project in the team, which really centers on the question of why Meniere's disease occurs. Meniere's disease has been associated with a multitude of factors, including genetics, environmental conditions and exposures, as well as some emerging evidence showing underlying cytokine profiles. And this is essentially talking about inflammatory conditions that may play an important role in developing this disease. In addition, recently there has been some evidence to suggest that epigenetics, and in particular DNA methylation, may also be playing an important role in the development of this disease. So understanding why this disease occurs at the molecular level is an essential part of the puzzle we need to research so that we can find better treatment and diagnosis options for our patients. So my project involves looking at whether DNA methylation can help solve any of the current questions we have regarding Meniere's disease molecular features. So what is DNA methylation? DNA methylation can be considered as an additional layer of information that the cells rely on to navigate the extensive information that is stored in the genetic code. For example, if you consider your DNA to be like a big encyclopedia, then DNA methylation can be considered like the bookmarks and tabs throughout the book, which would help us to read the pages of the book in the correct order. So at a molecular level, this looks like a chemical modification on the left here of the slide. A chemical modification that is attached to the C base of the ACGT that makes up the entire genetic code. The most important feature to remember about DNA methylation is that depending on where in the genome it occurs, it can suppress the expression of genes. So you can see here a gene that is switched on the left in green, gene 1, because it doesn't have methylation at the start or head of the gene. However, this gene 2 on the right is switched off because it does have methylation at the gene head. So what we can do is profile where the methylation is across the entire genome to understand patterns. Now, these patterns are normally present on all DNA, and it functions to help in various normal processes such as cell development and aging. However, what we also know is that many disease conditions have very abnormal patterns of DNA methylation, which causes the wrong genes to be switched on and off. And this dysregulated pattern can create abnormal molecular pathways to be active or inactive, which can in turn exacerbate the disease condition over time. We have very well characterized examples of this in certain disease contexts, such as cancer, diabetes, and Alzheimer's disease, to name a few. So one of the questions we would like to know is, does DNA methylation play an essential role in the molecular profile of Meniere's disease? As part of the team here at Colleen, we have a rather unique opportunity to analyze this problem. Professor Escamez and his team in Granada, Spain, over several years, have managed to gather a rare collection of Meniere's disease blood samples, which have been carefully extracted to provide DNA and RNA for various types of data collection, including WGBS, whole genome bisulfite sequencing. This is a specialized technique that allows us to gather information about DNA methylation patterns across the entire genome. That's every single ACGT in the entire DNA code. It's a lot of very valuable data. So we have whole genome DNA methylation information for over 70 healthy controls and Meniere disease patients, some of whom who also have vestibular migraine phenotype. Now you can see from these graphs, the majority of the DNA samples are from patients who are between 40 to 60 years of age, with more female samples than male samples. And this is quite reflective of the general population because Meniere's disease is often more pronounced in the 40 plus age group and is more likely to affect women than men. So over the next several months, I'll be investigating whether we can identify the significant differences between DNA methylation patterns in DNA taken from Meniere's disease patients compared to healthy people. Now you might be wondering, why is this important? Well, unlike genetic mutations, epigenetic modifications are known to be dynamic throughout one's lifetime. These patterns in particular change as we age, as we are exposed to different diet and lifestyle conditions. They change as we get sick from disease and we heal from disease. Not only that, but we already have some pretty good strategies that can artificially modify these DNA methylation patterns. For example, in certain types of leukemia, DNA methylation inhibitor is often used in the clinic to remove some of the unwanted DNA methylation patterns that co-occur with this disease. And this is really useful for cancer treatment. Therefore, if we identify that epigenetic modifications such as DNA methylation do indeed play a role in this disease, we have the opportunity to explore new treatment strategies which are currently not being considered at all for this condition. So if we were to realize particular gene or molecular pathways heavily impacted in Meniere's disease through epigenetic mechanisms, we'll be able to identify this through the type of analyses that we have planned to do in the team. So thanks very much for your time, and I will now pass it on to Dr. Prathamesh.
So I'll introduce myself. I'm Prathamesh Nadapanaya. I'm research fellow at Meniere's Disease and Neuroscience Laboratory. So I'd be explaining what I'm doing I just joined 2 months ago. I'll be talking about what I'm doing in the lab. So our plan is to establish a mouse model, animal model, to study the Meniere's disease. So what you are seeing in the left-hand panel of the screen is actually the specialized cells, which Antonio was just talking about a few minutes ago, inner and outer hair cells that are being stained by actin filament. So what we are actually seeing is the one row of inner hair cells and 3 rows of outer hair cells in the mouse organ of Corti, the apparatus, the structure, the tissue that's responsible for hearing. So it all started a few years ago when our team at the University of Granada found that mutation in the gene autogelin, which was, we assume, is responsible for the Meniere's disease. So it was found in 4 families, and the 2 mutations, which is V269I and VA2037, could possibly be very important for our study. So what we decided was, and before we start on it, we wanted to know what the gene did, which is the autogelin gene. So we knew that the autogelin gene is expressed in 2 very specialized tissue in the organ of Corti, which is the tectorial membrane and the outer hair cells, which again was Antonio was talking about it. So these tissues are so important for hearing function. So we thought, okay, why not we do a mutation in these? So our next step was to generate a transgenic mice line using standard CRISPR gene editing technology for these 2 mutations. Now the question arises, why we use transgenic mice? One of the main reasons is the hearing and vestibular inner organ in the human and the mice are very similar, very highly conserved. Secondly, using mouse model, we can do ultrastructural studies as part of the investigation, which is impossible in human patients. And thirdly, whatever knowledge we have so far about all the hundreds of proteins that are actually important for hearing function was studied in mouse model, meaning whatever knowledge we have was actually from these transgenic mice, which we actually applied in our clinical trial or human studies, whatever we are doing now. So it's very important that we use transgenic mice. So in short, we can say that mouse model for hearing research is the gold standard to study inner ear neurobiology. So for my project, my aim would be like to test first the hearing and vestibular function of these 2 mutations in the transgenic mice. To start with, to do that hearing study, we'll be testing using auditory brainstem response, which we call it in short ABR. So for this, we'll be placing the mice, anesthetized mice, in a soundproof chamber, and we'll be non-invasively, most importantly, there are no surgeries involved, and the mice can wake up in few minutes after the experiment. So we place the electrode non-invasively around 2 to 3 millimeter beneath the skin, and we place a speaker in front of it, and which will be playing white noise, white sound. And we'll be testing depending on how the mice reacts to this sound. So we'll be testing frequencies ranging from 8 to 32 kilohertz. You'll be surprised that the mice can hear as low as 4 kilohertz to 64 kilohertz, which is a lot as compared to a human, which could hear from between 12 to 28 kilohertz. But we don't have the technology to measure all the frequencies, so we measure which is accurately. So we measure only from 8 to 32 kilohertz. Second part of the hearing study we'll be doing is looking at the distortion product autoacoustic emission. So this actually is done to see if the outer hair cell of the mice is actually functioning. As I just mentioned a few minutes ago, that autogelin mice is also expressed in the protein is also expressed in the outer hair cells, meaning if the mutation causes it to become not active or dysfunctioning, we can know by this test. So this test is also again non-invasive. What we do is we place a probe non-invasively in the external ear canal of the mice, similar to how we put our earbuds in our ears for listening to music. And we play 2 tones from the speaker, which goes inside the ear, and the outer hair cell reacts by sending another signal, which comes out, which is being picked up by our microphone, and we can measure if the mice can actually hear, like if the outer hair cell actually is functioning. And the next one would be to do a vestibular function test, where we will be doing rotor test, which is very standard, which many labs do, which is really established. And this test is used to study the aspects such as motor performance, balance, and ataxia. Before I move on to the next part of the presentation, I want to remind you all about sound exposure and danger for the ear. So what we are seeing here is the infographic, which is in green, and green, red, and brown. So this is actually, the green is actually the permissible amount of noise intensity in decibel, which doesn't cause any irreversible or permanent hearing damage. Meaning, let's say till 70 decibel, which is like when you're using a vacuum cleaner, or it's like in a playground, noisy playground. So that's like permissible amount which we can hear, and it doesn't cause any damage. But when we go a bit higher, say 90 above or 100, it's like chainsaw or a gun or a plane taking off in close proximity, that can cause, or I would say would cause, irreversible and permanent hearing damage. So scientists have already done study in mice where they expose the mice to 100 decibel of noise for like say an hour, and they noticed that there was progressive bulging of the resonant membrane in the tissue inside the hearing apparatus and hearing loss. So when I say bulging of resonant membrane, it's actually it's actually the endolymphatic hydrops, which is also another what you call symptom that Meniere's disease patients have. But I'm not saying that noise actually induces it, but it could. That's our whole point. And the noise also causes inner and outer hair cell degeneration after this acoustic trauma. So what we hypothesize is that these two mutant mice, which we are planning to generate, would be more susceptible to noise-induced damage as compared to the wild type. Another reason we are hypothesizing is that the otogelin is also expressed, as I mentioned, in the tectorial membrane. So we assume that with this mutation, the tectorial membrane, which is in the organ of Corti, might be thinner, making it more susceptible or more vulnerable to noise-induced damage. So to test this hypothesis, to test the susceptibility of the mice to noise, we'll be exposing the mice to noise, say 1 or 2 dB for an hour. Then we'll be measuring the hearing function of these mice from 8 to 32 kilohertz. And we assume that the hearing threshold for the control would be really less, meaning they are unaffected, but whereas the mutant would have a higher hearing threshold, suggesting that the noise actually was more susceptible, the mice were more susceptible to noise, and they had a hearing loss or an increased hearing threshold. We are not sure, but that's what we are planning to find out. So to conclude, our team in University of Granada in Spain found two mutations in the Spanish population who had Meniere's disease. And we are planning to generate these two mouse models by CRISPR-Cas9 in Australia, in our lab at MDNL at Corning Institute. And we are planning to test the hearing and vestibular function in these transgenic mice, of course, as compared to the control. And we'll be testing the susceptibility of these mice, this mutant, to noise as compared to the control. I would like to end by thanking our team, all of us, like Professor Antonio, Vaiba, Christina, Dr. Jacob, our RA, and everyone else who are part of the team.